Now, media is used to grow microbes inside of the laboratory. In nature, microbes pretty much will grow on anything. But with media, we have the ability to dictate what bacteria are going to eat. And so we refer to this as artificial media or synthetic media because the researchers are the ones who end up making the media to grow their bacteria. This is an example of a media that's known as a triple sugar iron agar. And in this media, we have 3 different sugars that are added, as well as the element iron, because some bacteria can digest iron. In the media, we also have a pH indicator, which will allow us to know if the bacteria are actually using the resources and nutrients that we've included inside of the media. Now, there's different types of media. There's chemically defined media, and this is a type of media in which we know all the components that exist inside the media. Media can come in the form of either a solid or a liquid, and in this particular test tube we have a solid media, but you can also have media growing in liquid, such as Alexander Fleming had when he grew bacterial organisms using broth. And we typically just use broth as a liquid media inside the laboratory. An enriched media is a broth or a solid media that has been given a rich supply of nutrients in order to grow a specific organism. So some examples of enriched media are things like chocolate agar, which has some chocolate added inside of it. No, I'm just kidding about the chocolate agar. Chocolate agar doesn't contain chocolate in it, and in fact it contains red blood cells that have actually been lysed, and when red blood cells are lysed, they turn a very dark brown color, and so that's how you actually get the name chocolate agar. But there are some other enriched mediums out there where you can add things such as salt, such as mannitol salt agar plate, and salt will actually, that's the wrong example. Salt will inhibit the growth of gram-negative organisms like E. coli, but the mannitol sugars inside of the agar plate will actually enhance the growth. So in a sense, it can be referred to as an enriched medium. Another type of medium is that of selective, in which you add specific inhibitor elements that can cause the bacteria to not grow or discourage the growth of the bacteria. And some examples we have are agars such as that mannitol salt agar plate, where you have salt that inhibits the growth of the bacteria. Some other things are things like MacConkey agar or eosine methylene blue agar. And in these particular agars, they contain certain dyes that will inhibit the growth of gram-positive bacteria, which enhances the growth of gram-negative bacteria, and that makes the medium very selective. So if the medium is selecting for a specific group or a specific organism, then we refer to it as a selective media. Here is a medium that's known as differential medium, and differential medium essentially is medium that can be used to help us distinguish between different types of microorganisms. So you can figure out whether or not a microorganism is a cell respirator or a fermenter. You can determine whether or not a bacteria likes to digest glucose or lactose or sucrose. So differential media helps us to differentiate between organisms. And an example is that of thioglycate broth. With thioglycate broth, you can tell the difference between organisms that are obligate aerobes. You can tell the difference between organisms that are microaerophiles or the organisms that are considered to be obligate anaerobes. And this is based on where these types of bacteria grow inside the media. So obligate aerobes you would find growing at the surface of the broth. Microaerophiles you're going to find growing towards the bottom of the broth, but the organisms growing at the very bottom of the test tube are going to be obligate anaerobes because they can't tolerate any concentration of oxygen. And so in this test tube, you have a little bit of oxygen that's still present here at the top of the tube. But as the depth of the broth gets deeper, the percentage of dissolved oxygen gets lower. So at the bottom of this broth tube, there's no oxygen present. At the height of it, at the top of it, you have 21% dissolved oxygen. And so you, after 48 hours of growing your bacteria, what you'll find is you'll see clouds or turbidity of organisms growing here, growing in this region here, or this region down here. And that's how you'll be able to differentiate between the different organisms and their error tolerance properties. So it's very neat working with differential media. Another type of differential media is that of blood agar, which is simply sheep blood and agar mixed together. Agar is a solidifying agent, and then sheep blood is just the component that we add to the media. And what you can see when you grow some bacteria on this plate is that the bacteria will secrete a specific enzyme known as beta hemolysis. And that beta hemolysis enzyme is going to destroy all the red blood cells it comes into contact with. And so as bacteria such as Streptococcus, and Streptococcus is also a bacteria that resides in the oral cavity, once this bacteria starts secreting this enzyme, or this exoenzyme, the enzyme will start to break down the red blood cells. So if you can imagine growing bacteria on this plate, you can see bacterial growth wherever you have these colonies, and around the bacterial cells you see this halo effect. This helps us to know if a bacteria is considered to be hemolytic, for one, and if it is hemolytic, this will tell us how that bacteria will react in our own blood. So if a patient were infected with this bacteria here, and this bacteria got into the patient's cardiovascular system, that can be a very big problem, because look at the effect this bacteria is having on all the red blood cells in this plate. So loss of red blood cells leads to loss of oxygen, which can lead to death. So it's really important for us to grow microorganisms on blood agar, so that way we have a better understanding of how they will grow when they're inside of the body. Now, when we culture bacteria, we use special instruments called inoculation loops to isolate them and to grow them on our nutrient agar and differential agar, or different media plates. So what we have to do is isolate our specimen from a specific source. So if you're working in a clinical laboratory, you might have a sample of sputum taken from a patient, and you may have to purify that sputum so that way you can isolate the bacteria that might be present in it. And so to do this, what you can do is you can take a simple inoculation loop, take a sample of the sputum, and then plate that on some nutrient agar media. And you make sure this is completely sterile, and then incubate your plates for 48 hours at either room temperature, 25 degrees Celsius, or 37 degrees Celsius, or whatever optimal temperature growth it is you think your organisms you're looking for will grow at. So you do that inoculation, and then you have to wait. So the standard process that takes place when you do this is complete sterilization of the inoculation loop. So your inoculation loop consists of a loop that is on a wire. So you have to heat this wire till it's red hot to sterilize it. And you do that by passing it over a Bunsen burner flame. Once you let that flame cool, then you can collect a sample of your bacteria, and then you can spread it on your Petri dish plate. Wherever you touch this Petri dish here, as long as you have bacterial cells on your inoculation loop, you're going to get bacterial growth. There's really no way you can mess that up unless your loop is too hot and you went to collect your sample and then you killed the bacteria and it didn't transfer over. So you get your sample, and that's what's being demonstrated here from this plate. And then in this particular technique, which is called aseptic transfer, you're taking from a plate and you're transferring bacteria from the plate into a nutrient agar slant, or into some fresh media. So you collect your sample, insert your sample in the new media, and then you have to close your test tube with the enclosure, and then you're going to sterilize your flame again to make sure you get rid of contaminants that are growing on side of the inoculation loop. Okay, so this is actually a very simple technique, and it's employed all the time in laboratories that require culturing of bacteria. Now, once we have our plates inoculated or test tubes inoculated, the next step is to incubate them. So this is the same as incubating chicken eggs, or incubating bread in the oven, or incubating anything. What you do with incubation is that you let things sit at a specific temperature. So you can let them sit at the appropriate atmosphere, the appropriate moisture level, but most importantly the appropriate temperature, because remember, temperature is a big influencer and dictating and controlling microbial growth. So in this process, we typically grow bacterial organisms at either, as I've said many times, 37 degrees Celsius or 25 degrees Celsius. 25 degrees Celsius is room temperature, and 37 degrees Celsius is our body temperature that you would want to see if microorganisms grow at. So how do bacteria grow? Well, bacteria grow essentially on a growth curve. And remember, we're not talking about bacteria in terms of growth regarding width or regarding height, because essentially they're always the same size. When we talk about growth, we're talking about multiplication of bacterial cells, going from one bacterial cell to several thousands of bacterial cells. On this graph here, I'm going to get this text out of the way. There are 4 phases of bacterial growth. There's the lag phase, where essentially you have bacteria just kind of hanging out, getting ready to grow and develop, and right now they're in the process of maturing. But once they mature and they start undergoing the process of binary fission, bacteria will enter what's called the log phase. And in the log phase is where you're going to have massive replication, massive binary fission, and where you're going to have replication of the DNA, and then the formation of a septum inside of the bacterial cell, causing the cell to split into 2. And this is going to continuing and continuing and continuing and continuing and continuing until the species has met its particular incubation period that it's optimal for. So say bacteria, and most bacteria tend to grow logarithmically for 48 hours. After 48 hours, essentially what you'll see is the bacteria will start to level off, and they'll just kind of refer or go into what we call the stationary phase. In the stationary phase, the bacteria are just kind of there, and they're existing, and they're eating nutrients. They've already replicated. They might replicate again. But in stationary, stuff is stationary. It's like, oh, well, we're just going to hang out now. And then eventually, as all things in life, we have the death phase. So once all the nutrients and water resources have been depleted in the environment the microbes are living in, it's time for them to die. And so these are the 4 phases of microbial growth and bacterial population. And this whole diagram here is referred to as a growth curve. Okay, so again, bacteria grow on an exponential growth curve. That's why we have this referred to as the logarithmic growth phase. And then they enter the stationary phase, and after stationary phase, they enter the death phase. Okay, and so once that takes place, we have the end of the bacteria. Okay, so let's talk a little bit about inhibiting the growth of microbes in vitro. We've already talked about enhancing the growth of microbes. Now we want to talk about how it is we can control their growth by keeping them from growing too much. So the first thing that we do is we simply disinfect and we sterilize. The key thing in the laboratory setting that helps us to control and minimize bacteria from causing outbreaks after studying them in laboratory is to make sure that all of the media that has been used and all of the contaminated materials that have come into contact with bacteria get sterilized. And so to sterilize these items, we place them in a giant system known as an autoclave. The autoclave is very useful because we can insert our instruments and then heat them to an incredibly high temperature to destroy organisms.